Hi everyone, Miss Lerner here. I'm here to go over and review some difficult concepts from Unit 4, the Unit 4 review activities. Hopefully, in watching this video, you can ensure some common error pathways that you may have had and correct them in order to get 100 on the accuracy assignment that is due on Thursday at 8 p.m. Okay, so let's start with it's sensational. Everywhere there's a blank, I was looking for some sort of term. Okay, so starting with vision. Light is reflected through the cornea. It goes through the pupil, okay, which is the hole that is going to be controlled by the most beautiful muscle in your body, the iris. That light will hit the lens. The lens will change shape. C for change. There's C's in this term accommodate. Now, accommodate, or when the lens accommodates its shape in order to reflect the image on the back of the eye, you should have heard that word in another context before. Accommodation in development is when you change existing thought structures or schemas. Okay, so the lens is going to shift shape. The light will then be placed back, and the image will be flipped and inverted onto the back of the eye, the place where the magic happens, which we call the retina. Okay. Now, on the retina, the first kind of layer of the retina that is going to be responsible for where the magic happens, transduction, is going to be composed of the photoreceptors. Now, the photoreceptors were rods and cones. Okay. So cones were responsible for clarity and color and fine detail. They are concentrated on the fovea, which is the center cones of the eye. If you want to take a good look at something, it's right in front of you. Excuse me. And rods are responsible for vision in darkness and dim light, peripheral vision, because they're not in the center of the retina. They're in the periphery of the retina. Remember my shady Uncle Rod hanging out in the darkness, spying on me in the periphery. Okay, so this right here, cones concentrated on fovea and rods, fire. Okay, so I'm going to go over to the associated theories part because that goes with what we're going to talk about with the way that cones are grouped in the fovea and the retina. Okay, in the retina, cones are going to be grouped in three types of clusters. Cones that are sensitive to red wavelengths, blue wavelengths, and green wavelengths. Okay, essentially all colors are going to be a combination of these three colors. Okay, red, green, and blue. So when you actually overlap red and green, you get yellow. Like think about an old TV screen, the pixels, and how that works out. Okay, so the first theory, three types of cones in retina for blue, red, and green. That was a theory that was called the Young-Helmholtz trichromatic theory. Now, you could just say it's a trichromatic theory, three colors, that's fine, okay? This explains the earliest stage of color vision, that first layer in the retina. Now, if enough cones fire, it goes to the second layer of specialized neurons. These specialized neurons are known as bipolar cells. You can see here, they activate the bipolar cells. Bipolar disorder is from one extreme to the other. Bipolar cells, likewise, are going to be grouped in complementary pairs. Red-green, yellow-blue, black-white, okay? When someone sees a red wavelength, that red side of the bipolar cell is going to fire, and the green side is going to be suppressed. And then if I stare at something for red that is red long enough, and then I look at a white piece of paper, the green is going to come flashing back, okay? Because then the green overcomes and the red is then suppressed. This is what is known as the opponent process theory of color vision, okay? But we'll come back to that. Well, I guess I could just put this right here. Opponent process theory. Okay, so again, I've written it down here, the red, green, blue, yellow, and black, white sensory receptors in the bipolar cells. when one sensory receptor, should say, is stimulated, the other is inhibited, and it says explains blank and color blindness. Well, I just said what it explains. It explains after images and color blindness. Okay, so when someone is colorblind, like the most common type is red-green color blindness, and it's, they see everything in kind of this muted, kind of tan mauve type of way, and that's because of a deficiency within the bipolar cells. If enough bipolar cells get activated, then the message moves on to the next specialized layer, which is that gang of lions. Remember when I used to draw it on the board and there were like clusters of lions? Ganglion cells make up the optic nerve. Okay, so it's the last layer because it's literally going to form the optic nerve. Then it's going to go through the thalamus all the way. So it says impulses sent to the thalamus, then sent to the blankety blank, think optical. Okay, that's going to be the occipital cortex. I shouldn't say occipital cortex. I should say occipital lobes. The visual cortex is in the occipital lobes. It sets off blankety blank. These are neurons in the visual cortex that respond to different types of visual images. Feature detectors. Feature detectors, when I see certain things, like I see a person's face, there are going to be specialized cells that are going to fire in response to seeing eyes and a nose and a mouth, and then it's all going to come together in a specific part of the brain known as the fusiform gyrus, and it's going to say, oh, there's Miss Lerner. Okay. Just to kind of reiterate with this, because usually when we talk about theories, they're in contrast to one another. These two theories of color vision both must exist to explain the entirety of color vision. So the trichromatic color theory explains the first stage of color vision, okay, and the first layer of the retina where the cones are clustered, okay, and then from the bipolar cells onward, vision is explained by the opponent process theory of emotion. And I've written that in a very, in caps and star. Color vision can only be fully explained by combining these two theories. Okay, so moving on, hearing. Okay, by far, like the most studied and researched senses are vision and hearing. Okay, so the other ones, we have a little bit of terminology, but we really need to know the exact progression for vision and hearing. Okay, so these are energy senses. So light waves are energy, sound waves are energy as well. So sound waves, a physical energy wave, collects in the pinna. These are our pinnas, okay? They're funnels that allow us to localize sound. If I only had one ear right here, I wouldn't be able to tell where you were talking from, okay? They travel down the auditory canal. They hit a blank, okay? The common name for what they hit, a boom, boom, is the eardrum. I'm really looking for the more formal name, tympanic membrane. It vibrates against the three smallest bones in your body, which we are known as a whole as the ossicles. There's the hammer or malleus. There's the anvil or incus, and the stirrup or stapes. Okay, and these are just called that because that's what they actually appear like as bones. They rub up against the entrance to the cochlea, which is called the oval window. Okay. The most important thing for you to understand is that the cochlea is where transduction happens in hearing. Okay. If I were to unravel the cochlea and look at the floor of the cochlea, I would have these hair cells like swinging around in the breeze. Okay. They are transducing physical energy into psychological impulses to the brain that are going to be interpreted as sound. Okay. The hair cells move and transduction occurs. Impulses travel via the auditory nerve. Yeah, the auditory nerve through the thalamus and then to the temporal lobes. Okay. Temporal, think it's like by your ears right here. They're responsible for hearing and smell and memory. And there are two theories that explain how we hear pitch with hearing. And both have to exist to explain how we hear very high pitches and low pitches. Okay, so the place theory says that there are different locations or places in the cochlea for different frequencies of sound. Okay, so, and this is how we explain the high range of pitch. The frequency theory is going to say that pitch results because of hair cells firing at different rates in the cochlea and the auditory nerve. The rate at which the cells are firing is different, not the location of them. And this explains low pitch. Okay. Moving on. Touch. Okay. There are a variety of aspects of touch: pressure, temperature, pain. Okay. This is interpreted in the brain in the somatosensory cortex. The somatosensory cortex is going to be right in the front of the parietal lobes. It backs up to the frontal lobes and the motor cortex so that if I feel something, I can respond to it very, very quickly because they're so close together. Endorphins, the brain's natural blank, are involved in controlling pain. Opiates. I can be your hero, baby. I could take away your pain. Don't you remember Enrique Endorphin singing that song? All right. Now, there's one theory associated with touch, specifically pain itself. Okay. It says that there's essentially two pathways that sensation can travel on. One is a more slow-moving pain pathway, and one involves all sorts of stimulation. And there's going to be a gate from the spinal cord to the brain to interpret that information. So if I stub my toe, the first thing I do is I run around the room kind of screaming, ow, 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 ow. What I'm doing is trying to get the stimulation on the fast track to beat out the slow-moving pain message on my slow track to shut the gate so I don't feel the pain, and the pain is delayed. Okay? Because of this sort of rush to the gate, this theory is called the gate control theory of pain. Right. So for this reason, like sometimes when people have mosquito bites or something, they'll put like an X in it, and it really makes it less sort of itchy and painful. Okay, gustation. Okay, well, gustation deals with the fact that we have these taste buds, and underneath these taste buds, we have the fungiform. The taste buds are located on these mushroom clouds known as the fungiform papillae. The taste buds enable kind of the liquid to taste a chemical sense. This is a chemical sense, no longer an energy sense. Taste is going to relate to chemicals and molecules. Taste and smell are going to be very much interrelated. If you're blindfolded and your nose is all stuffed up, and you eat a raw apple and a raw potato, they're going to seem like the same thing. It's hard to believe, but true. Okay. The fancy term for smell is olfaction. It is we believe it is the oldest sense evolutionarily. It is the only sense that does not pass through the thalamus. Okay, so it says here chemicals picked up by receptor cells in the nostrils, which are linked to the olfactory bulb and go straight to the amygdala. And you could either write limbic system, because it's, or you could also write the hippocampus and the hypothalamus. Why can I not type right now? Okay, so that's why smell, you have like that knee jerk, sort of you smell something, your stomach might drop, or you might be transported right back there because it doesn't have that pathway, that thalamic pathway where it's a way station where it has to be routed. It just goes straight there. And that was evolutionarily viable. because smell was very dangerous for us. Okay, our sense of smell may be a powerful trigger for memories because, speaking of which, we are conditioned from birth to make strong connections between smells and events. The nerve connecting the olfactory bulb sends impulses directly to the limbic system. The receptors of each nostril connect with the cortex. Smell is a powerful cue for encoding memories into long-term memory. Strong smells encourage us to process events deeply. This is the answer, right? That the nerve connecting the olfactory bulb sends impulses directly to the limbic system. Okay, the cochlea. Cochlea is where the magic happens in hearing. So protects the surface of the eye. No, they're trying to trick you. That's the cornea. Transmits vibrations received by the eardrum to the anvil, the hammer, anvil, and stirrup. Not so much. Transforms vibrations into neural signals. That sounds pretty good. Coordinates impulses from the rods and cones. No, not wrong sense. Sends messages to the brain about the orientation of the head and body. No, that the orientation of the head and body is the vestibular sense. That would be the semicircular canals in the ear. So I agree that it is in the ear, but it's not the cochlea. So it is transforming vibrations into neural signals. The blind spot in our eye. So we all have a blind spot. We saw it in class where we had the X and the O and one would disappear. But because of the Gestalt principle of closure, we have expectations of our environment and we actually fill in those blind spots at all times. But why is there a blind spot? Okay, well, it says here it results from, what are the possibilities? The lack of receptors at the spot where the optic nerve connects to the retina. The shadow the pupil makes on the retina. No, it's not like the shadow. It's not like the pupil has like this huge shadow in your eye. Competing processing between the visual cortices in the left and right hemisphere. Floating debris in the space between the lens and the retina. That would be scary. Retinal damage from bright light. This is because there are no photoreceptors where the optic nerve connects to the retina. So you're going to be blind in those areas. Okay. What is the principal difference between amplitude and frequency in the context of sound waves? Okay, so going back to cover the correlation between physical energy and psychological sensation, okay, for sound waves, amplitude is the height, right, of the wave, and that is going to translate into loudness. Frequency is the wavelength or the number of times a wave completes a cycle in a given space, and that is going to be pitch. Okay. As far as light goes, it has a similar kind of relationship. The amplitude of a light wave is its brightness, how bright something is. Whoa. Okay. The frequency or wavelength of a light wave translates into its color or hue. Okay. So, amplitude is the tone or timbre of a sound. Frequency is the pitch. Nope. Amplitude is detected in the cochlea. Nope. Amplitude is the height of the sound wave, while frequency is a measure of how frequently the waves pass in a given point. Yes. Okay, there we go. Which of the following statements best defines the gate control theory of pain? Pain impulses are sent to receptor sites in vital organs. Non-painful nerve impulses compete with pain impulses to reach the brain, creating a neural blockage. Stimuli of various kinds activate free nerve endings. Pain is simply a psychological state, not a physical one, or perception of pain depends on one's physical makeup. It was this. Non-painful nerve impulses are going to compete with painful ones. You're shown a picture of your grandfather's face, but the eyes and mouth are blocked out. You still recognize it as a picture of your grandfather. Which type of processing best explains this example of perception? Bottom-up processing, signal detection theory, top-down processing, opponent process theory, or Gestalt replacement theory? Okay. So signal detection theory deals with how apt you are to notice the stimulus, depending on your expectations or motives. So that is not it. Opponent process theory is color vision. Gestalt replacement theory is fake. So it has to either be bottom up or top down. And I'm going to say, by and large, this is one of the most confusing topics for students to understand. Okay. Top down processing. I want you to think about it like from the brain down. It's your expectations, memories, culture, context, and it's going to color your perception of an event, okay? Bottom up is looking at those specific features and building then an interpretation, okay? My memory of my grandfather's face is going to enable me to immediately, even when the features are blocked out, know that that is a gestalt of my grandfather. So the answer here is top-down processing. You're not just saying, oh, it's a face, it has eyes blocked out and mouth blocked out. That would be bottom-up processing. You say, aha, that's my grandpa. Then you know. All right, monocular and binocular depth cues. There are a ton of monocular depth cues, which just mean that if you were blind in one eye, you would still be able to see them, see depth perception. Artists are going to really, really prey on these depth cues. There's only two, count them, two binocular depth cues that are going to involve both eyes, okay? The two binocular depth cues are going to be retinal disparity and convergence. Retinal disparity just means that you have slightly different images from each eye that come together to give you depth perception. So if I put my hand out here and I look at my finger, I close one eye and the other, my finger should dance. If it's closer, even closer together, my finger should move even more, okay? Because that's how the retinal images compensate to give you that depth, okay? The other binocular cue is what is called convergence. That my muscles are going to converge on a object far away, and I'm going to feel a certain amount of musculature that is looking to focus on that object. And then if I go closer, it's going to be even much more intense. Okay, so this one is asking which of the following depth cues could you not use? So you need a binocular depth cue. Texture gradient, no, that's a monocular cue. Like we look at fine textures being really, really close up. We're convergence. Well, I just said that. Okay, convergence, yes, is a binocular depth cue. Linear perspective. This just means like when the railroad tracks go into the distance, that you can see because they come to a point that it's going farther away from you. Interposition. If I'm behind something and you can't see me, you know that that thing is in front of me. That is another monocular depth cue. And shading. We constantly shade when we're in art class because that is a monocular depth cue. Color blindness and color after images are best explained by what theory of color vision? Opponent process. That was on the chart. What behavior will be difficult without our vestibular sense? Integrating what we see in here, writing our name, repeating a list of digits, walking a straight line with our eyes closed, or reporting to a researcher the exact position and orientation of our limbs. The answer here is D. Why is it not E? Why is it not reporting to a researcher the exact position and orientation of our limbs? Well, I'll tell you, because that's a different type of sense. The vestibular sense is going to involve the semicircular canals, and the fluid in the semicircular canals makes it so that you're balanced and you don't fall over. The kinesthetic sense is going to be your ability to have coordination, okay, that you're a good athlete, a good dancer, not a klutz, okay? And the receptors for the kinesthetic sense are in your muscles, tendons, and glands, okay? So very different. Okay, which of the following best describes the relationship between culture and perception? Okay. Our perceptual rules are inborn and not affected by culture. I'm going to tell you that is completely false, okay? We have what are called learning-based inferences due to the culture that we grow up in, okay? And it is going to color our perceptual world, okay? Does that mean that if I am in a very different culture that I'm not going to necessarily see things the same exact way? Well, they're going to have slight changes. Certain optical illusions that one culture will fall prey to, another one won't. So there are certain optical illusions, like the Muller-Lyer illusion, that have one line, like two lines, right? And they're both the same length. The top line has lines going this way on the edges, and the bottom has lines going this way, okay? In Western cultures and cultures that grow up with having corners in their houses, even though you might know that these lines are the same length, it's really difficult to take away the influence of having corners that you grow up with. And you're always going to say that the bottom line looks longer. Now, if I grew up in the Hutu tribe in Africa, and I grew up in a hut and there were no corners, I'm going to say that those lines appear and are equal every single time. Okay, so really, most perceptual rules are going to be the same. Okay, but some can be varied. Some can vary slightly. Okay, so let's see if there's one that is going to come out here. Most perceptual rules apply in all cultures, but some perceptual rules are learned and vary between cultures. Slight variations in sensory apparatuses among cultures create slight differences in perception. No, it's not like if I were born in Australia, I have like different ears or something. The processes involved in perception are genetically based, so genetic differences among cultures affect perception. Nope, culture is really a construct. It's not going to be like genetically based. Okay. 11. Taste, 1 gram of table salt in 500 liters of water. Smell, 1 drop of perfume diffused in a 3-room apartment. Touch, the wing of a bee falling on your cheek from 1 centimeter away. These are all examples of just notable difference. Difference threshold, absolute threshold, adaptation of our senses, or the perception of our senses. The answer here is an absolute threshold. An absolute threshold is the minimum amount of stimulation you need to understand that there is something there 50% of the time, whether it's light or sound or taste or touch. Okay. So I'm looking out in the distance in the absence of electricity, and half of the time, according to psychophysical researchers, from 20 miles away, I should be able to say there's light in the distance from just one flame of a candle. Okay. That is the absolute threshold for light. Now, just noticeable difference or difference threshold are used interchangeably. They are going to signify how much, whether you notice a change when there's already a stimulus. So instead of noticing that there's light at all, you walk into a room and you say, is it brighter in here than it just was? Okay. Or you turn up the volume on the TV and you say, can you turn it down? That's way too high. Okay. Wow. Now, the difference threshold or just noticeable difference is going to be proportional to the intensity of the stimulus. So if the stimulus is very loud to begin with, you're going to have to turn it up a lot in order to notice a change. If it's very, very quiet, you only have to turn it up a little bit, a little click to notice a change. Okay. This is what is known as Weber's law. Okay. When the molecules of a skunk spray enter your nose, the molecules are transformed into electrical signals or impulses that are interpreted by the brain as an unpleasant odor. This is an example of transduction. It's the magic. Getting sprayed by a skunk. Magical. My dog got sprayed by a skunk this year, and it took like 3 months for it to fully go away, even after bathing him in tomato sauce. It's beautiful. All right, so Weber's law comes back. Okay, Weber's law. Let's see. Is it the smallest change in stimulation can be detected 50% of the time? No, that's just the just noticeable difference or the difference threshold. Is it the principle that the JND for any given sense is a constant proportion of the stimulation being judged? That sounds really good. The principle there's an adjustment of sensation levels depending on the stimulation received. The theory that the least amount of energy detected in a stimulation only occurs 50% of the time. No. Or the theory that all stimuli respond to the same sensations through the process of creating meaningful patterns. That's Gestalt. Okay, so yeah, the principle that the just noticeable difference is very high when the stimulus intensity is high and low when it is low. That is what B is saying. And 14, as a car drives away, it projects a smaller and smaller image on your retina. Although the retinal image grows smaller, you do not perceive the car as shrinking because of shape constancy, size constancy, shape continuity, or size perception. Okay, we all have perceptual constancies. Shape, size, okay, color, and that's what the blue and black and white and gold dress deals with, like the fact that your color constancy is messed up in that one specific scenario because you're using the backdrop of your own expectations, and depending on whether you see the dress outside or inside, you're either going to see it as white or gold or blue or black. So when the retinal image is growing smaller, but you're not like, oh my gosh, that car is shrinking, that's going to be an example of size constancy. Similarly, if a dog is running toward me, I'm not going to be like, oh my god, exploding dog. All right. So I hope that this was helpful. If you have any questions, always reach out. I'm hoping that you have a better understanding of some of these concepts. I'm also hoping that you really have enough stamina to finish this year out well. I'm gonna do my darndest, and I'm sure that most of you will too. I miss you all. Please check in live sessions if you can. I think it's a really good time to interact with your peers. Good time to interact with me. I'm a little bit selfish. I kind of would like to see you, but you could always turn your camera off. You could always just come and mute yourself. But I would like to see more of you. All right. Have a great day. Bye-bye.